So Leah, do you want to do your advertisement or
Sure.
notice now or at the end when more people are on?
It's up to you. I was going to have Heather put in the link. Maybe I could state it now, Bob, and then if there's time at the end, you can kind of just reiterate. But just to let folks know, on October 21st and 22nd, we will be kicking off a series of meetings that focus on biotypes of CNS complications in people with HIV. It's kind of comprised of 5 different working groups with absolutely stellar speakers. Bob Heaton is one of them. Scott Latender, I don't know if he's on here yet. Trish Berto. And so it's really to start thinking, as we all know with CNS complications, there's a ton of heterogeneity, it's complex. And so trying to pull us all together. And I can say just from going into these working group meetings, the discussion I think is going to be really great. So please register and send that link to as many people as you want, because we want tons of people to attend. So I think that's it, unless Bob, you have anything else to say?
No, that's
but this good. Yeah. And so Bo Ansis and I have been spearheading this with NIMH. And I should also say that, NIDA, NIA, and NIMH, they're all kind of integrated across all of the working groups. So I think, yeah, it's free too. Thank you, Heather. So with that, I will hand it over to you.
Okay, thanks, Leah. So today I'm subbing for Norm Howie, who at the last minute I guess couldn't make it today, and we want to welcome our speaker, who is Gary Siuzdak, and he is professor and director for metabolomics and mass spectrometry at the Scripps Research Institute. His lab facilitates a range of biological and chemical research and has developed important new methods for data acquisition and data analyses. Gary's talk today will be entitled Activity Metabolomics Enabled by METLIN 4D. Thanks for being here, Gary.
Okay, thank you, Bob. And yeah, this has been a really interesting week at Scripps. And as a result, I've actually altered my talk a little bit as a result of that. I mean, I was originally going to talk about a couple of different things, and I've decided to remove one aspect of it and place in some of the original pain work, kind of in honor of the work that Artem had just won the Nobel Prize for. And interestingly, this morning, Ben List, who won the Nobel Prize in chemistry this morning, he originated here at Scripps as an assistant professor and did some of the original work with Richard Lerner and also Carlos Barbas on this effort. So it turns out that I just got off the phone with her. One of my staff members originally worked with Ben and worked on some of the proline catalysis work that he originally did, and I believe was the part of the organo catalysis work that the prize was awarded for. So it's been an exciting time here at Scripps, and hopefully I'm going to be able to reflect some of the things that are going on here, not only in terms of the analytical types of work that I'm involved with, and I'll jump into the talk now. I changed the title of the talk a little bit. It's called Activity Metabolomics Enabled by XCMS METLIN. These are the technologies that we've been developing over time. As Bob alluded to, I run the Center for Metabolomics and Mass Spectrometry. So mass spectrometry is one of our primary technologies that we use to perform our work. And I consider myself more of a technologist both in training and in terms of what I like to pursue for research. But unlike, I think, most technologists, we really rely very heavily on our collaborations and the biology that we're performing with our collaborators to help guide us in terms of what we're developing as technologists. So, and I hopefully will give you a flavor for this today. I'm going to be covering a large number of topics. I mean, so here are some of the main papers associated with it. Basically, it'll span roughly 30 years of actual research work that we've been performing. However, the reality is I'm only going to have two simple messages, both one towards the middle and one towards the end. And I think these messages, at least for me, resonate quite strongly in the types of things that we're pursuing here at Scripps, and also with respect to what I've been doing over the last 30 years. So the first one is this work that we did in the early 1990s with Richard Lerner, and it involved the sleep process. It was a really very simple study. It was we were looking at sleep-deprived animals and looking at their cerebral spinal fluid and looking for molecules that were showing up more strongly as a result of sleep deprivation that could potentially have some therapeutic value, could potentially be used as to understand sleep a little bit better. And this is where we developed, was kind of the initiator for what we ultimately developed, which is XCMS, which is the technology that allows us to compare LCMS data to pull out molecules that are significantly dysregulated within these samples. So XCMS right now is, since its origins many decades ago, it's actually been, or a couple of decades ago, it's actually been garnered a fairly large user population. We have now over 50,000 registered users on XCMS. It's largely a metabolomics platform. And basically, in its essence, it allows you to do comparative analysis of peaks that show up in the LCMS chromatograms. And actually, what it's capable of doing is, in its original approach, it was the original approach to nonlinear align both LCMS and GCMS data. It was also the original approach to do statistical assessment of dysregulated peaks. And since then, we've now incorporated a systems biology aspect to it, and also use it heavily to ultimately identify the active metabolites in a sample. So in that original effort that we were working on with respect to sleep, we saw hundreds, if not thousands, of different features in these samples that we're analyzing, the cerebral spinal fluid. And we did see a set of molecules that were significantly dysregulated. And as a technologist, and what we did was ultimately try to identify what these molecules were. We used accurate mass measurements, tandem mass spectrometry data, which basically fragments the molecules and creates a pattern that helps you identify them. We used HD exchange experiments, preparative LC to collect them, the metabolites for NMR, and then ultimately synthesized an authentic standard to test for its activity. And the molecule that we found the most interesting ultimately is this molecule right here, oleamide, a fairly simple fatty acid amide molecule. And very encouragingly, what we found was that the molecule oleamide induced a sleep-like state in roughly 4 minutes after administration. So clearly it was an interesting molecule. However, very discouragingly, at least for me, is that it took about 8 months to identify what the molecule was. And again, you can see it's not a very complex molecule, so it should have been much easier. But I think that was more a reflection of the state of metabolomics at that time. There were no databases with respect to with empirical data in them on these molecules and the metabolites. And we really were up against many different obstacles in terms of doing this sort of identification. So this actually created the challenge to our lab to identify more effective methods for identifying metabolites or molecules in general, and to do that actually fairly rapidly. And that's where METLIN comes in. Actually, METLIN originally developed, and we originally developed it in the early 2000s. And it's been undergoing a pretty dramatic change over the course of the last 2 decades since we originally created the database. It was built after the sleep study to facilitate molecular identification, and currently now it has what I like to think of as METLIN 4D, or 4 different dimensions to it. The first dimension is the M/Z, so basically just getting mass measurements on the individual molecules, and that's the first dimension. And basically that gives us elemental composition. And back in the early 2000s, it was quite common for people to identify molecules just based on this elemental composition information, which unfortunately turns out to be fairly inaccurate. I mean, because there's a lot of molecules that are isobaric and will have the same elemental composition, but completely different structures. I mean, a good example of that would be glucose. So if you go into METLIN right now, you'll get roughly 200 hits for the elemental composition of glucose. So you get lots of different molecules that have the same elemental composition. So clearly, I mean, this is a good first step, but it doesn't give you the key information that you need to start getting more rigorous identifications. What has been really key to this has been the tandem mass spectrometry data within METLIN. METLIN has by far the biggest set of MS data, empirical MS data on molecular standards. And I defined the MS data as the fragment intensity and the precursor intensity versus the M/Z. And here's an example, that oleiamide molecule. This is the precursor, the intact molecular ion, and these are the subsequent fragments that have been generated in the tandem mass spectrometry experiments to give you this characteristic fragmentation pattern, which allows you to do these sorts of identifications. So we began with roughly 100 molecular standards in terms of generating this tandem mass spectrometry data. And since 2018, we've had significant growth in this area. And this gives you a kind of a sense of the growth that we had. It was gradually growing over the course of roughly 15 years or so, and then it really took off in 2018. And now we have, the number is actually 860,000 molecules within METLIN. It really is it's safe to say it's a fairly astronomical number in comparison to the other databases that are out there. We've been populating it fairly rapidly, and it's grown to really a very big, an approach or a technology that allows individuals like myself to, within seconds, provide a very relatively good identification on molecules when we see them within the database. So some of the statistics, and again, I'll go through this fairly quickly, but I do think it's relevant because it is at least for the metabolomics field, I think this is a fairly important set of information. We have 860,000 molecular standards, each with MS data. Each of those molecules has been analyzed at 4 different collision energies in both positive and negative ionization modes. And our success rate is actually roughly 74%. So not every molecule we're analyzing has been successful for a variety of reasons. They ultimately fail our filters because of low intensity or a variety of other reasons. So this 860,000 molecules that we have successfully analyzed is actually the result of analyzing well over 1 million molecules. So obviously, we're in a really nice position in terms of our automation capabilities here at Scripps, and this has facilitated the acquisition of this sort of data. And also, METLIN has over 350 different chemical classes of different molecules in it, so it really is quite diverse as well. And I always like to acknowledge places like Cayman Chemical and Avanti Polar Lipids for facilitating the growth that we've had in the database, among many other places and organizations. To give you a bit of a context for this, I mean, here's METLIN at 860,000. The next biggest is the NIST database at 31,000, and then there's an array of other databases that are out there. This sort of information may be less interesting to you, but from a personal perspective, what I'm about to show you, I think you will find perhaps interesting. This is my lab, or a portion of my lab, and you can see there's mass spectrometers in there, and there's just a host of, we have roughly 22 mass spectrometers in the center. These are just some different views for different sections of the lab. So this next one is particularly interesting because this is a section of the lab that we actually caught on video camera just prior to when we underwent that really significant growth. So if you look at this, it's a normal lab. We have, I think, let's see, 1, 2, 3, 4 instruments in this portion of the lab. But in 2017, I was on vacation. I got a phone call, and it turned out that the lab actually had roughly 10,000 liters of distilled water dumped on it. And obviously at that time, it was quite devastating, and it wiped out a whole bunch of our mass spectrometry technologies. And it was something that took a while to recover from. But ultimately, it turned out to be useful in that some of the instrumentation that we had, namely this particular instrument and others like it, we were able to replace them all, and also actually buy some of them back from the insurance companies, fix them, and ultimately that became the heart of our METLIN technology. At least the other interesting thing I like to bring up is that, from a perspective of working at this for so many years with METLIN in particular, working at it for about 20 years now, that all of the data on METLIN right now is on this little chip right here that I have. So when you think about it from a career perspective, that everything you've done with respect to a major product can be put onto something like this. I've always found that fascinating, though. It's perhaps a little bit sobering as well. The other couple of aspects of this that I'm just going to go through actually quite quickly has been 2 recent developments, things that haven't even been published yet, and that's the METLIN neutral loss database. This is a completely new database that looks at neutral loss spectra as opposed to tandem mass spectrometry data. And it gives us a completely different perspective on the mass spectrometry data. You can see the data actually looks very different. Tandem mass spectrometry data looks like this. This is a complete version of the data. And the question is, why would we want to use this, or why would we want to create a neutral loss database? And the answer is actually quite simple. Here are 2 molecules. They're almost identical, except in this molecule you have a hydroxyl group. In this molecule you have a ketone, and otherwise they are identical. But when you look at this tandem mass spectrometry data, there's virtually no overlap except this one particular ion. However, the neutral loss data actually does show significant more similarity. So we're using the neutral loss data to identify similar molecules that are not yet in the database. And this is still a big issue in metabolomics in terms of identifying unknowns. So we think that the neutral loss spectra is going to be a very key aspect to accomplishing this. This is a more extreme example from a therapeutic perspective. This is the rosuvastatin, which is a statin, and this is the desmethyl version of it. This is the MSMS data. The only difference between these 2 molecules is that with the original molecule, it has a methyl group right here, and the desmethyl version has the methyl group missing. So otherwise, these molecules are identical. But again, you see there's very little overlap in the tandem mass spectrometry data. However, when you look at the neutral loss data, it gives you much more accurate picture of the similarity between these molecules. So again, this sort of information is going to be, I think, key in the future for helping us do these very difficult identifications, which is very typical now in metabolomics. I gave you the statistics before for METLIN tandem mass spectrometry data. Interestingly, there's no databases right now for neutral loss. So I think that'll change pretty soon, but it's quite interesting to me that nothing exists at the moment for these sorts of comparative analyses. And then finally, and we're going to use this to actually do we're combining both the MSMS data and neutral loss to ultimately create a machine learning algorithm, which we've already created to help identify or perform more accurately the similarity analyses between molecules. And then finally, the fourth dimension, which is a relatively new technology for the lab. We have two instruments that are performing this right now, both a Bruker timsTOF and also a Waters QTOF instrument that has ion mobility capabilities. And we're creating a database of a very orthogonal piece of information, which is ion mobility or CCS values to do these identifications. So obviously we've been harnessing our automation to create the METLIN CCS, and ultimately why are we doing this? And the answer is that it just creates another dimension in identification integrity. And where we've already leapfrogged some of the other databases, we're now at about 30,000 in terms of ion mutability data versus some of the other smaller databases, which hover around 3,000 or so. Okay, so that's most of the technology aspect of this. And I think the next part perhaps is what you'll hopefully find even more interesting. And that's on the biological side. And it's really been the biology that has driven everything that I've spoken to you before. And I think from this group's perspective at Johns Hopkins, this work that I'm going to show you now is something that I did with Howie Gendelman and Mike Bosca a couple of years ago. And actually, I first wanted to answer that first simple message that I mentioned at the very beginning. There were two ultimately simple messages. The first simple message is that empirical data is a driver for metabolomics, and the bioinformatics is the key to making this data valuable. And that's what I hope to show you with respect to the biology in a few seconds. So what is the second simple message? Well, I'm going to get to that at the very end of this presentation. And I'm about halfway through right now. And the first example that I'm going to show you is in the neurodegenerative work that we did with Howie Gendelman. This slide is a little bit older, but I like to use it. It just gives you a sense for the fact that this is still a very prevalent disease, as I'm sure you're all very well aware of. At that point in 2015, 37 million were currently infected. And but what I'm going to focus on is that the neurological disorders are common during the course of the disease and typically referred to as neuro-AIDS. And this is what I focused on with our work with Howie and also with Mike. And I also like to point out that Mike passed in a glider accident a couple of years ago. So. But he was really key to making this a successful study. So what we did in this particular study was we looked at the free dolutegravir or free DTG, and it has been one of the most widely used treatments for HIV infection, and we compared the brains of individuals who were not only treated with the free DTG, but also the nano or the nanoparticle approach that Howie had developed and is still developing under a variety of different names, but he's still developing the nanoparticle-based approach for delivering this and other drugs. In its current form, or at least in the form we used it in, the particles are about 20 nanometers in diameter, And the beauty of it back then was, and now I think he's increased the time scale significantly, is that it allowed patients, or the idea was to allow patients to only receive a monthly dose of the drug instead of the daily dosages that you typically get with the free version of the molecule. In our case, we used a mouse model, and we looked at different sections of the brain to see how the animals were responding to the drug. And of course, as I mentioned earlier, that the technology that we used to perform these analyses was XEMS, to analyze the LCMS data that we were generating from the different sections of the brain. And we did comparative analyses between the different sections of the brain in both the free version of the molecule and the nanoparticle version of the molecular delivery approach. So here's the free version of data coming from the free version of the dolutegravir administration versus a control where the animals were only given a vehicle. And what you're seeing here is on the top portion here, we have the molecules that have been upregulated in the animals that have been administered the free dolutegravir, and on the bottom portion we have molecules that are downregulated. The size of the bubbles that you're seeing here indicate the fold change, and the color intensity. The darker the color, the more statistical significance, and this is typically related to the p-values associated with the statistical assessment of each of these different metabolites that were being observed, that were again upregulated here and downregulated here. The retention time is on the x-axis, and this refers to when these molecules were coming out, and on the y-axis is the m/z. So you get a sense for the size of these molecules. In this particular case, they're ranging typically anywhere from a couple of hundred Daltons to typically under 1,000 Daltons. So that's a fairly typical representation of what we see when we do these experiments. When we looked at the nanoparticle delivered DTG, actually we were seeing a very different profile. The first thing that jumps out at you with respect to this profile is that the molecules there's much fewer molecules that are being dysregulated. From a metabolism perspective, it's pretty clear that the animals are not being as impacted metabolically in the brain as they were with the free DTG. So what we set out to find was what was the predominantly underlying cause for this significant difference between these two, the significant difference between these two administrations of the nano DTG versus the free DTG. And what we ultimately found was that there was a high level of consistency with respect to the oxidative stress pathways being significantly dysregulated especially in the animals that had been given the free DTG. So clearly the free DTG was actually causing a higher level of oxidative stress than the nano version of the delivery. And here's a couple of so we ended up focusing in on a couple of different molecules to hone in on this particular observation. So I have everything is compared to the control. We have the free DTG versus the nano DTG. And again, the control levels are shown here in black. And here we have the ascorbic acid actually going down in the free version versus the nano version. The nano version actually is slightly lower, but not nearly as low as the free version. And actually, this was even more predominant with glutathione, which is a great indicator of oxidative stress. Here you see the glutathione was clearly being significantly impacted by the free DTG, whereas with the nano version, there was still an impact, but not nearly as prominent as what we were seeing with the free DTG. So, I mean, there's a couple of reasons why what we were observing with the nanoparticles was different from the free version. One, I think there's two reasons this could be happening. I think the one is that the nanoparticles are basically suppressing the reactive oxygen species that are being generated, and as a result, reducing the oxidative stress. So that's one possibility. And the second, perhaps more obvious one, is that the slow release of DTG is causing less oxidative stress. There is precedence for this. I mean, that it has been shown in a number of studies that these nanoparticles can reduce the generation of reactive oxygen species. So, and just from a practical perspective, it's not unrealistic to think that the slow release of the DTG could be also a reason why we're getting less oxidative stress. But the combination of these two is probably the reason why we were seeing so much less oxidative stress. I mean, my understanding, and again, this is not my field, but my understanding is that the nanoparticle delivery area is still undergoing significant development, and there's significant challenges associated with it. But this, I think, is one indication of why it could potentially be a useful delivery method. While we're waiting for that, though, and this would fall into more of a speculative part of the talk today, I'm reminded of another molecule that causes significant oxidative stress, and that's Tylenol. I mean, if you overdose on Tylenol, if you take too much because you're trying to manage your pain, of course, you can cause significant liver damage. And one of the molecules that we, from a clinical perspective, that we turn to reduce the oxidative stress is N-acetylcysteine, or NAC. And for all the clinicians out there, I'm sure you're very familiar with this. N-acetylcysteine is essentially considered a drug that ultimately results in the formation of increased glutathione production, which helps to reduce the oxidative stress in the liver and is the go-to drug for acetaminophen overdose. I would argue that it's perhaps an underused molecule simply because it could be used in, again, this is the speculative aspect of this study, but it could be used to potentially reduce the oxidative stress from some of these free these drugs like DTG that clearly are inducing some oxidative stress. It's so what you might ask is there any reason to believe that this could be impacting oxidative stress in the brain in terms of this drug? Could it impact oxidative stress in the brain? And the answer to that question is yes. There's been a number of studies that have been performed with N-acetylcysteine, double-blind, placebo-controlled studies specifically on schizophrenia, to determine if the N-acetylcysteine could reduce glutathione levels in cerebral spinal fluid, and the answer has actually been yes. The glutathione replenishment does reach into the brain. So again, from a purely speculative perspective, I think the types of oxidative stress type drug molecules that side effect could potentially be alleviated using things like N-acetylcysteine. The next part of this, my presentation, or the next biological story I'm going to tell you about relatively quickly, is on the story of pain that we've been involved with. And this was motivated for me. I was motivated to tell you about this today because of Artem's work. His was more of a mechanical study. This one has turned into more of a chemical or metabolic study into pain. And what we were interested in doing is, we know that chronic pain or neuropathic pain afflicts millions of people worldwide. Available treatments are still relatively ineffective, as we can see in the headlines today with the different companies that were originally promoted. Some drugs that are highly addictive, and our objective was to try to get a better understanding of what's taking place during this process of pain. And so we wanted to identify and potentially verify novel pain pathways using our metabolomic-based approaches. The experiments that we did was on a mouse model, was we were looking at what is called tibial nerve transection, where we cut a nerve and they'll let it heal. And it's well known that these animals continue to have pain or sensitivity even after the wound has, by all appearances, been solved. The healing process is complete, but the animals still have sensitivity. So it's a classic example of neuropathic pain. So what we did was we took our platform, we did our untargeted metabolomics to do the comparative analyses, we did the data processing to pull out the molecules that were most interesting in terms of statistical significance and intensity, and then we did targeted validation to further validate that what we originally observed in the untargeted experiments was real and could be validated with a more robust sort of system. And again, the typical experiments with XCMS is you do the comparative analyses across different LCMS runs, look at molecules where there's a significant change, and then ultimately, hopefully with METLIN, we identify the molecules typically through tandem mass spectrometry type information. Okay, so I think that I'm willing to open up this particular part to anybody who wants to jump in and answer this question, because I love asking this question, because the answer to it actually surprised me. When we originally did these experiments, we looked at all different portions of the animal. We looked, of course, where the tibial nerve transection was taking place. We looked at the plasma. We looked at where the nerve was entering the spinal cord. We looked at the brain. We looked at many different areas. So the question I have for you is, and we saw lots of changes taking place across the system. Where do you think we saw the biggest changes occurring in terms of the sheer number of metabolites and the intensity of the metabolites that were occurring? Okay, so actually my answer was at least my personal answer to this was the tibial, where the actual surgery took place. And actually it turned out to be absolutely wrong. I mean, we didn't really see very significant changes taking place here. In the plasma, which actually we were kind of enthusiastic about, in terms of the observations, what turned out to be basically very little to no changes occurring, which may not, in retrospect, may not be that surprising, simply because the plasma has such a dilution effect that it ultimately wouldn't give you, it'd be very difficult to pull out the molecules that were significantly changing. The brain too, I was expecting to see some changes there, but again, the dilution effect is so big that we didn't really see any appreciable changes taking place. It turned out that the place where we saw most of the changes were taking place at the dorsal root ganglia, where it entered the spinal column.
Actually, Katie Riggs guessed that one, Gary. She put it in the chat. Katie Riggs, she guessed it and put it in the chat box, so she didn't get the credit she probably deserved.
Okay. Yeah, no, because I have to say I did not guess that when we first did these experiments, and I was actually quite shocked to that's actually where it was, where the change, most of the changes in. And who's the person who guessed it?
One of our
Katie
trainees.
Riggs.
Yes.
All right, yeah. So, and she was. This pie chart actually is just. It shows how right she actually was. I mean, this pie chart actually represents how, comparatively to the other areas where we were looking, how big the changes were and how many metabolites were actually being dysregulated. So it's kind of an all-inclusive sort of pie chart, but the difference was really quite significant. I mean, ultimately, we ended up focusing in on a couple of molecules that we thought intuitively made sense that could be involved in this process. So we again, you could spend years, if not decades, on this sort of project to ultimately deciphering what's actually going on. So we had to use some level of intuition to try to decipher what was happening, and ultimately we came down to a couple of molecules that were particularly interesting in terms of their structures. Here we have dimethyl sphingosine. At the time, this molecule was a novel molecule and had not been previously identified in animals prior to this. The other molecule, again, just based on elemental composition and accurate mass measurements, the other potential candidate was sphingosine C20. And again, based on the elemental composition, steroyl ethanolamide was another candidate. And so based on accurate mass measurements, you really can't distinguish these because they have the exact same mass. However, when you compare the tandem mass spectrometry data, it became quite clear that it was the dimethyl sphingosine was the molecule that we were actually looking at this particular process. Okay, so for me, this is where, again, from a biological perspective, this is where it got interesting. I mean, you can look at the dimethyl sphingosine is the one we ultimately focused on. There were other molecules that we didn't focus as much attention on, but from a pathway or a mechanistic perspective, it turned out that they were ultimately interesting with respect to the tibial nerve transection experiments. Partly because with the ceramide molecule, which we were also seeing dysregulated, it could act as a precursor to the dimethyl sphingosine. So we decided to follow up with this on the animals to see what impact the dimethyl sphingosine was having on the animals. The previously unknown as I said, it was a previously unknown metabolite, and again, the dimethyl sphingosine was having an impact that was very similar to what you'd expect with capsaicin, which is the active ingredient in chili peppers. And you may have heard of capsaicin this weekend because it came up in the as one of the molecules that was being used to ultimately identify the receptors for pain in the Nobel Medicine prizes this weekend. So DMS was actually providing a response that was similar to the capsaicin. And also in terms of pain withdrawal threshold, which is a common technique used to identify the impact of a particular molecule on an animal in terms of its sensitivity. It's called the pain withdrawal threshold, and it measures the relative force needed to induce pore retraction. Here's a normal rat. Basically, by poking the animal with a needle, it basically doesn't experience any significant pain, or its pore withdrawal threshold is relatively high. However, the animals that were treated with the dimethyl sphingosine, they had a relatively low poor withdrawal threshold. So it was, again, this was an indicator that the DMS was actually being, was active in this neuropathic pain. If you look at from a mechanistic perspective, the DMS is the pathway to forming it would naturally go through ceramide, as I mentioned earlier, and through the ceramidase enzyme. So what we became interested in was, could dimethyl sphingosine be ameliorated by introducing an inhibitor to ceramidase, or finding what was a term coined in the 1950s as an anti-metabolite, basically an inhibitor to the enzyme. And it is known that N-allenyl ethanolamine is an inhibitor to ceramidase. So what we decided to do was test the N-allenyl NOE to N-allenyl ethanolamine against in the animals to see if that had any impact on the animals that have been administered the DMS, and also the animals that had the tibial nerve transection. So here we have the controls, as I showed you earlier. These are the injured animals, and here we have the injured animals that have been treated with the anti-metabolite NOE. So clearly their poor withdrawal threshold actually improved quite dramatically when after a couple of days after being administered the NOE. So that's the end of that story. So I've basically given you 3 stories so far. I mean, I've given you, well, maybe 4 if you consider the technology part. I've given you the story about technologies and how we've been developing the technologies to help us identify metabolites. But I've also talked a little bit about sleep. I've talked a little bit about neuroaids, and I've also talked about neuropathic pain. So what is the second simple message? And I think for those of you who are interested in what I'm about to tell you, these are 2. This is a perspective in Nature Biotechnology that we had on this last message, and here's this is a Nature Reviews paper that both of them I'm particularly proud of, because I think they actually go into the use of metabolomics and how where the areas where it can be particularly most useful. So what is the second simple message? Well, I'm going to answer that question with respect to what we typically consider the central dogma of biology. Typically with biology, you think of DNA going to RNA, going to proteins, and going to metabolites, and ultimately the metabolites reflecting the phenotype reasonably accurately. And that's why we spend so much time, or a lot of metabolomics people spend so much time trying to identify biomarkers from looking at the metabolites. But of course, metabolites interact with, and this is very much appreciated, is that they interact with all levels of biology. From a DNA perspective, they interact with in terms of gene expression, epigenetic regulation, and signal transduction. From an RNA perspective, you have ribose switches, and you have post-transcriptional modifications. And of course, from a protein activity perspective, you have allosteric regulation, and you have catalysis by cofactors, and also post-translational modifications. So what my final message isn't that that this is new information, that metabolites can be actually acting on these different levels, because this is well known. Nobody is going to say this is new information. What I think is interesting and relatively new is the fact that we have a technology in place that will now allow us to more effectively identify metabolites that haven't been previously associated with these protein or RNA or DNA interactions. And I think, as I mentioned in the previous studies, all 3 of those studies, the molecules we ultimately ended up focusing on probably would not have been the first molecules you go to try to understand what was taking place with respect to those. Is there a precedence for this? Actually, obviously, we've been doing this for quite a while, since the early 1990s. And in 2018, I was looking at this fairly closely, and you can actually see a number of studies that actually demonstrate how different molecules, this one at top with taurine, we actually demonstrated its impact on multiple sclerosis and myelin regeneration. Just in 2018, there were a wide variety of different papers that came out in different journals that really speak to the fact that these small metabolites can have a significant impact at regulating phenotype, and metabolomics can have a really nice impact on identifying them. So the second final message is metabolomics is typically thought of in terms of biomarkers and pathways, and that's what we typically focus on. What I would suggest is that it should be more focused on identifying metabolites that modulate physiology. Given this is, it's a more time-consuming process, but I think with the pathways and biomarkers, the information that you're getting out of there is more, I would say, almost housekeeping. Whereas when the identifying metabolites that modulate physiology is, it's, at least for me, a much more exciting process, because you really can see these molecules potentially interacting with systems and potentially altering in a very positive way. And I can give you some examples of this, of why this is an important area. I think I mentioned statins in a very different context earlier, but it's known that statins, of course, are great drugs. They lower the impact of arterial sclerosis, the risks associated with that. But one of the negative things associated with statins is that it lowers CoQ10 levels. And CoQ10 obviously is involved in the in how your muscles function. So and this is now prescribed by physicians. If you take your statins, it's common that you're also being prescribed CoQ10 to supplement the loss of CoQ10 via the statins, again, to alter any potential side effects that you may have from statins. And there's other examples of this as well, but I think that is a nice one to kind of end with. So with that, I just want to acknowledge and I think I'm right on time in terms of what it was allocated for me. I want to acknowledge the NIH, the DOE, and the DoD for funding. There's been many different labs that have provided molecules for us. As you can imagine, with 860,000 molecules, it really is there's been a lot of different contributors to this. Most recently, it's been Cayman Chemical and Avante. And again, actually, interestingly, I did a quick calculation of just the contributions that Avante and Cayman have made, which has been on the order of thousands of molecules. If you look at their price list, the value of those molecules is roughly $3 million. So $3 million in free contributions of chemicals is really quite generous on their part. The individuals involved with this, Winnie Heim and Aries Ains-Forna, have been really prime movers in making METLIN what it is today. And the sleep work was originally done by Richard Lerner and Ben Cravat. And actually, it's probably worth noting that one of today's Ben List winning the Nobel Prize today, I think is to a significant extent based on a paper in 2000 in the Journal of American Chemical Society on proline catalysis. And what's interesting about it is that Richard was, there were only 3 authors on the paper. Ben was the first, Richard was the second, and Carlos Barbers, who passed about a decade ago or so, he was the last author on it. So it's kind of nice to be able to mention that in terms of this acknowledgement of Richard, in terms of this original sleep work, which on his part, with respect to the sleep work, I really this is one of the first examples of people using metabolomics to identify molecules that modulate phenotype. So, and Ben was instrumental in creating the synthesizing the oleamide molecule. And I already mentioned Howie and Mike in terms of the neuroaids. Oops. And Oscar and Gary and also Caroline were all very instrumental in the neuropathic pain study that I mentioned. And that's it. I'll be happy to answer any questions. I can go to the chat if you like, or if people want to just ask.
Gary, I don't see any. Thank you very much for a really fascinating talk. I don't see anything in the chat box. Would anybody like to just speak up?
Dr. Latender, I think you had a question.
Well, it was a while ago, but it's okay. I actually, Dr. Espinosa Kai responded to me about some of their work, but I just back when you were presenting the data on the free dolutegravir and the nanoformulated dolutegravir, I just was wondering whether you measured intracellular concentrations to see if there was a dose effect response over time, or whether that just didn't need to be done because Dr. Gendelman's probably long since done that work in this particular cell type with those particular formulations.
Yeah, so again, it was a while ago that we did these analyses, and I to be honest, I can't recall how many different we actually looked at multiple different animals, and if I'm trying to recall right now, I believe there were multiple time points where we saw the most significant changes with the free versus the nano versions was in the initial time points, and of course, as you'd expect over time, the that oxidative stress impact would diminish as the drug went away. Again, I do apologize. It's been at least 6 years since I've done these analyses. So I do recall that we did do some time course work on it. Again, what I showed you today was perhaps was nicely representative of the general impact that we're observing of the nano versus the free. And again, most of you are probably very much more in tune with what's going on in the field right now. And I'd be curious to see if you think my speculation that something like a NAC prodrug that could boost, and it's a fairly benign prodrug that could boost glutathione levels in the brain, could that be used not only with dolutegravir, but also other drugs that are known to cause oxidative stress to alleviate that well-known side effect?
What little I know about NAC is that it has a favorable safety profile, which is what we would care about the most in terms of putting it into humans. In one of our large studies, the Charter study, which is based at 6 sites around the US, we're seeing evidence now, in work much less elegant than yours, just measuring end products of oxidative stress and counting mitochondrial DNA, that indicators of mitochondrial injury and dysfunction and higher levels of end products of oxidative stress, including protein carbonyls and 8-hydroxydeoxyguanosine, being associated with things like depression or low amyloid beta and CSF, or higher volumes of abnormal white matter in the brain, even with effect sizes that are larger than sort of the typical inflammatory mediators that we look at. So I think and many of these people are treated with integrase inhibitors, and at the time this was done, probably mostly dolutegravir. So we haven't looked at a drug effect in those data. We tend to focus on whether someone is their viral load is suppressed, whether they're on effective therapy. But there's a pharmacology component of Charter as well, and we have generated drug concentrations in blood and spinal fluid in those participants. So it would be, the data are there. We're still sort of processing the pharmacology data, but we could look at whether there are relationships between higher dolutegravir concentrations, for example, extracellularly in CSF, and these end products of oxidative stress that would lend further support to the idea of giving something, safe like NAC to people when they're, while they're taking their antiretrovirals to protect them from the tissue injury that can result from excess oxidative stress.
So I know this presentation was recorded. So if you go back to the I think the 5 different references that I had in that in the middle when I discussed the dolutegravir and the NAC, and the 5 references refer to a schizophrenic study that was, again, a double-blind placebo-controlled study where they were looking at cerebral spinal fluid. And I was totally unaware of this, and this study, it was done out of the University of Lausanne, and I was giving a lecture there, and the study, the leads on the study came up to me and described how they started using NAC as a way to alleviate some of the symptoms that the schizophrenic individuals were having, because they found in schizophrenic individuals that the glutathione levels were low. So it was it's a really interesting, I mean, it's hitting on some of the things that you just mentioned in terms of, and so you may want to look at that to see if it has any relevance to what the thoughts.
Thank you. To your knowledge, are there any data in the Alzheimer's literature? That would be another big area of focus to sort of add it on again to people with Alzheimer's as a protective, easy to administer and safe intervention.
Yeah, it's interesting because I started myself supplementing with NAC after I've heard a lot of this anecdotal type of work, and also it's so that may seem a little crazy, but I've been given the potential benefits of it. I've been, but with respect,
I'm taking it as well, just to reassure you.
Sure. It's interesting because I have gone to individuals who do Alzheimer's type work, and not from the perspective of NAC, but more to use our metabolomics based approaches to see if we can find out something different than what is typically observed with respect to the amyloid peptides and such. And I think up until recently, I've gotten very little traction, but now there seems to be a more of an interest in pursuing it from a metabolomic perspective. And I wouldn't be surprised that one approach to work on this is just adjusting oxidative stress levels. But, yeah, thank you. I appreciate your comments.
So I guess we're out of time now. Very interesting discussion, and I'd like to again make a plug for the NIMH 2-day program, October 21, 22, that's chaired by Leah Rubin and Bo Ansis on biotypes of CNS complications in people with HIV.
So Bob, if you don't mind, I'd like to just jump in and answer one of the questions that was
Sure. Yeah.
Because I am it's one that's near and dear to my heart. The question is, I wondered, with the 850,000, how were they obtained, and how did we capture the data on them, and how many of them were endogenous, and is there any rationale behind selecting those molecules? My answer to that question is I basically have tried to get as many molecules as I can, both endogenous, exogenous. We have, I think, almost all the drug molecules and as many drug metabolites as we possibly can. I'm interested in small molecule peptides as well, because we see these in these studies as well. I'm interested in toxicants. So the short answer to Pragni Deem's question is We are trying to bring in as many molecules as we can into METLIN to make it as comprehensive as possible, because I think a lot of the things that we observed aren't just these endogenous small molecule metabolites. They range across toxins and all sorts of other different molecules. So thanks for giving me the time to answer that question, Bob.
Sure. Thank you again for a very stimulating presentation, and I hope you'll come back.
Yeah, thank you very much.
Okay, we'll see you all in, I guess, 2 weeks because of next week, the NIMH presentation that's causing us to stop for that week. So anyway, we'll see you all soon, and take care.
Okay, thank you. Bye.
Thank you. Bye, Gary.